Hello, I'm Jeremy Fry, Director of Sales at ProImmune, and in this presentation I'll introduce you to MutaMap, a mutational activity map to enable superior protein engineering decisions. MutaMap is a pioneering, high-throughput in vitro assay service for understanding the affinity or activity impact of point mutations in protein therapeutics, including antibodies, that enable superior protein engineering decisions. Molecular evolution techniques, such as phage display and other phenotype-genotype coupled randomization techniques, are most commonly used in the affinity maturation process for monoclonal antibodies and other binding scaffolds. The advantage of these technologies is that they help explore a very large sequence space of combined mutations. There comes a point, however, when final decisions have to be made on the implementation of a protein sequence where individual point mutations may be considered in an antibody or therapeutic protein to meet a variety of design objectives. Randomized molecular evolution is not appropriate for this step. Exploring individual point mutations is not a new approach, but it has been difficult to carry this step out in a very high throughput way, especially where the objective is to clone and express every mutant and then measure its affinity and activity with reasonable accuracy. This is what MutaMap can achieve for you. So when it's time to make your final engineering decisions for your antibody, MutaMap evaluates the effect of substituting each amino acid at each position in a protein sequence one by one, with all 19 possible substitutions, for example in the CDR regions of monoclonal antibodies. To do this, we first take the parental sequence and through a process of site-directed mutagenesis, protein expression using in vitro translation, followed by highly sensitive multi-point solution equilibrium titration immunoassay, we then generate a map of all the beneficial, permissive, or harmful mutations in the sequence of interest. Most importantly, a full characterization of 2,000 mutations, all 19 amino acid substitutions in a 100 amino acid protein sequence, takes just 12 weeks. To introduce the service to you in more detail, I'm first going to show you some data analysing the anti-VEGF monoclonal antibody Avastin. From this parent antibody, 5 amino acid substitutions were introduced to develop a higher affinity anti-VEGF antibody called Lucentis. The graph here shows the direct comparison of these two antibodies in the solution equilibrium titration assay, or SET, measuring the affinity of binding to VEGF. As a method, SET is particularly well suited for high throughput, high affinity interactions, unlike surface plasmon resonance, SPR, where the higher the affinity, the longer it takes to achieve a measurement. Here we present, in the form of a heat map, the MutaMap for Avastin. Data shown here focuses on the CDRH1 and H3 regions of the antibody. The parental Avastin amino acid sequence is shown vertically on the left-hand side. Horizontally along the top are listed all 20 amino acid variants. With regards to the key, the yellow colour indicates KD within threefold of wild type. This is a neutral effect on affinity. The green colour indicates an increased affinity compared to wild type, better than threefold. Orange, a poorer affinity than wild type, greater than threefold, where a red colour indicates no measured affinity at all, or the protein was not expressed. As you can see in the horizontal plane, there are key rows in red where mutations are clearly not permissive. An example of this is the glycine residue, 3 amino acids from the end of the CDRH1 region. Looking across this row, any residue other than a glycine at that position leads to a substantial reduction in affinity, shown in red. Other notable substitutions include the histidine to proline, which is the third residue down in the CDRH3 domain, which results in a marked increase in affinity. An insertion of a proline is not a mutation that would normally be rationally made. I'll now drill into more detail by looking at a single position. Serine 105 is one of the residues that is mutated in Lucentis. Here we plot each amino acid mutation against specific affinity measured for VEGF. In general, mutations at this position are well tolerated, shown by the high number of yellow variants. Only 4 substitutions result in reduced affinity, with serine to proline being the worst. Interestingly, the Lucentis mutation serine 105 to threonine is not the highest affinity variant. The substitution of a valine at this position actually results in a higher affinity for VEGF. So to conclude, MutaMap allows you to make informed protein engineering decisions for a range of key developability objectives, including cherry-picking which point mutations are permissible or beneficial in your protein sequence, including a quantitative determination of the impact on affinity. Combined with deimmunization approaches such as functional ProMap T-cell proliferation assays, it allows you to maintain or even improve protein activity whilst reducing the risk of unwanted immune responses. Since a key element to MutaMap is the confirmation of the ability of the protein to be expressed, the assessment of manufacturability and stability and other developability attributes are central to the advantages that MutaMap brings. Your antibody can even be engineered to improve the characteristics of species cross-reactivity for the purposes of preclinical safety testing. We can prolong half-life, and of course the whole process also generates unique new composition of matter with respect to intellectual property. Not only can single-point mutations be assessed, but of course we can also combine multiple mutations as a second round to confirm the synergistic effects of such design changes. And finally, turnaround time is an unbeatable 12 weeks for 2,000 mutants from start to finish. If you would like to learn more or request a quote, please contact us at the email address shown here or by telephone. We'll be delighted to hear from you. Thank you very much for your attention.